Press record. So today we got Carson Andorf from Maize GDB and lots of exciting developments that I've heard of over at Maize GDB. So I'll turn it over to Carson so we can start hearing about those.
Great. Thank you, Matt, for having me today. Reshare this. Yeah, so today I'm going to kind of share a project. This is in collaboration with Jeff Rossabara and Matt Hufford and Arun Seathram with MageDB on generating a variant data set and using this and incorporating it at MageDB to be able to explore the genomic landscape of maize. So if you're not familiar with MageDB, I'm assuming a lot of you might be. I just have one slide here, kind of just giving an overview of what we are at MageDB. We're a community driven. We have long-term federal funding from the USDA ARS, and we're a community database, and we provide informatics services to researchers focused on the important crop and the important model organisms via maize or maize. We have three major components. We have the community database. There's been a community database for maize for probably a little over 30 years now in one form or the other. We've supported over 20 different data types over the years. Our current focus is mostly on genetics, genomics, and breeding type of data sets, but we do have phenomics, transcriptomics. We're getting more and more epigenetics type of data sets. And over the past couple of years, we've started to incorporate some more proteomics type of data. On top of the community database, we build tools and resources to be able to access that data and visualize it and integrate it. We develop internal tools, but we also have third-party tools like genome browsers. We've developed some comparative genomics tools. We have BLAST, and more we're trying to use AI and machine learning to kind of supplement and complement some of the resources we currently have. And thirdly, we're the community hub for the maize research community. We provide services to like the Maize Genetics Cooperation, the Maize Genetics Meeting, and other training and outreach opportunities. So one of the big focuses at MaizeDB is the genomics. So we host a lot of the maize genomes at MaizeDB. We currently have 104 genomes that we host. This timeline and chart here kind of just shows the history of hosting maize genomes at MaizeDB, starting back in 2008 and 2009, when the first maize reference assembly was released. This was a historically important line B73. And until about 2015, we were a single genome resource until a few other maize reference quality genomes were released. And then we started to build some multi-genomic tools. In 2020, the nested association mapping founders, so these were 25 founder lines of that mapping population, were all sequenced and assembled and annotated using the same methods with the same supporting evidence data sets and using the same metadata. So that really allowed us to start doing some pangenomic type of work at MageDB. And in the past few years, the PanAn project, we're hoping to incorporate that into MageDB and start building these super pangenomes where we can look at all across Zia or the Andropogane clade and start doing some kind of super pangenome type of work. But the reference genomes, even if we have 104 of them, doesn't always tell the complete story. Maize is a very diverse species. There's tens of thousands of public maize accessions out there. There's even more probably in the public or in the private companies. So I just did kind of just a quick history of maize genotyping or resequencing. I'm sure there's some data sets that are missing here, but I just kind of want to show kind of how these projects, had different number of accessions they were looking at, the resolution, going back all the way to the HapMap projects. This is with Ed Buckler's group with some of these initial genotyping projects, and also with his group using the GBS technology. And you can see like HapMap 3 at about 1,200 accessions at 83 million coverage. We're using GBS technologies. They could look at over 60,000 different maize accessions, including about 15 or 16,000 public accessions at a 1 million resolution. And there's some other more targeted or specific projects that are looking at some targeted set accessions. Another one that's kind of noteworthy is last year, James Schnabel's group from Nebraska Lincoln uh, looked at about 1,500 maize accessions at 46 million. So it's kind of a general, including some accessions that weren't resequenced before. And this column here too just kind of shows the version of B73 it was mapped onto. And I kind of want to partly show this table just to show the difference of what each of these different projects are and the challenges that it presents to a group like us at MaizeGB in when trying to present these or integrate these type of data. So a lot of the methodologies and data formats can be different. They can use different techniques to actually do the variant calling, using different thresholds to filter it or different threshold criteria. Even the formats, a lot of these are saved in VCF, but VCF formats can be quite different on how the actual data is stored in them. And that makes integrating and merging this data really difficult. So we've hosted this type of data before at MaizeDB. We've had downloads, we've had genome browser tracks, we've even built tools to be able to search and query it. But they're usually pretty siloed, right? Because it's hard to make an apples to apples comparison with these. So a lot of these data sets end up being very separate from each other and almost impossible to merge this type of data. Another challenge is that the data can become obsolete or outdated pretty quickly. As you saw, some of these were done on earlier versions of B73. And even though we can do some kind of mapping, you know what I mean, maybe looking at flanking regions around a locus. But what happens there is as genome assemblies get better, even small little regions, like small little repeats, can get approved upon. So it's usually not all the regions are identical. So we either have a very conservative mapping criteria and we lose some of the data, so the coverage gets lost. Or if we want a high confidence set, then we continue to keep a high comfort, or if we want a more high coverage set, sorry, we can loosen up the criteria, but then we're losing the quality of the data. So really the best way to do this is just a full reanalysis of the data, taking the raw data sets, merging it together and remapping it to the genome of interest. But the challenge here is very resource intensive. It takes an HPC system, dozens, if not 100 of different nodes running for a long period of time. You need some level of persistent storage to store all the intermediate files and merge it at the end. So what we were trying to do is, this was Jeff's idea of coming up with a method where we can have a regular release of a large WGS or variant data set that we can release on a semi-regular basis, whether that's once a year, twice a year, you know, once every 2 years, something like that. But something where we have a regular release using the same method, incorporating the latest data sets that are out there. So this was kind of a pilot project for that larger idea. And we looked at some local resources, but we ended up working with Genco for this initial release, with the idea of looking at initially about 15 maize accessions. We want to include inbred land races and teosintase. So we want to look at some of these heterozygous SNPs too. We looked at different pipelines, but kind of decided on the BWA MEM SAM tools pipeline. And as I just mentioned, we also, including SNPs, we want to include indels and also be able to host heterozygous SNPs. So this is the initial data set we decided on. As I mentioned, about 1,500 maize accessions. On the left here is an accession count for each of these projects. So we chose about 10 different projects for various reasons to make sure we have a diverse set of maize accessions. We got to make sure there's high quality sets that are good read counts on these. So these are the bio projects at NCDCBI, and here's just a short project name of them. And as I mentioned, there's, we include land races. There's accessions from Canada, United States, Mexico, South America, Europe, China. Just trying to capture kind of a global and diverse set of maize accessions for this initial data release. So what we did, we gave these accessions to GenCove, they were able to do the variant calling, oops, for us, and we were able to download the data. So it was a really large VCF with no initial filtering. So we kind of wanted to be able to offer a large data set that others could do their own filtering on, but also where we can kind of look and control some initial filtering steps. So what we did, the first filtering step, essentially we looked at mapping quality greater than 30, coverage greater than 50%. So we had 1,500 maize accessions. So at least 750 or more of them at least had one read count at each locus. At the step, we removed multi-allelic sites. Then we did annotation of the variant effects using SNF on the latest B73 RefGen V5 gene model annotations. So I was looking to see if these variants were in intergenic regions, or if they were in the genes, are they exon, intron, are they synonymous, missense? Um, so just doing a full annotation on these variants. Uh, from there, we did a second filtering step where we use Planck to calculate the linkage disequilibrium, or LD, at looking for a max R squared of greater than or equal to 0.5. Just make sure there's at least one other locus in the data set that's highly linked to the variant of interest. So what we ended up with initially is kind of a high coverage set. This is one after the filtering step one, and a high quality set after filtering step two. We're planning on releasing these as VCFs. So if you want these data sets, they'll be available through download at MaizeGB. We also converted these to HDF5 databases. So this is something that Ed Buckler's group did for some of the HapMap and GBS data sets. It's a really great way to index this type of data, where you can kind of do some hyper chunking of the data based on the genomic regions. Lets you quickly access this data and attach a lot of metadata. So we can attach these filtering criteria. We can attach the annotations. Obviously, we attach the alleles, if they're the reference or alternative alleles. But we can quickly search this data using low memory, and we don't need a HPC to be able to query these database, which allows us to build a web-based tool, which is one of the ultimate goals, at least for MaizeGB, to be able to provide better access to this data. Then here, just quickly, here's the variant effect annotation table. As you can see, there's a lot of loci for this. So there's 228 in the high coverage, or 228 million for the high coverage, and a little over 75 million for the high quality. We'll likely maybe do some more filtering just to get a kind of a more manageable data set for some other purposes, depending on how you want to use this data. As you can see, a lot of it's intergenic data, but there are several million loci in the genic regions. One thing I want to point out here is the 1.3 missense mutations. So these are variations or mutations that actually cause amino acid substitution in the protein, and we're going to incorporate that into a tool we have at MaizeGB, and I'll show a demo of that in a little bit. So let me switch my sharing here and show you a prototype of the tool we're building at MaizeGB to get better access to this data. So some of you may or may not be familiar, but we had a tool called SNPversity at MaizeGB, which basically allowed you to query a VCF data set based on the genomic region, and also a subset of the accessions that were used to build it. So basically what we did, we just kind of had the same functionality of the original SNPversity, but we kind of just started from scratch. We wanted to make it fast and lightweight, so it's we're using JavaScript just to make it a front end tool that's a little more lightweight than it was previously, and allows us to be a little more flexible in incorporating new data sets in the future. So what you see here, this is kind of the prototype of this tool. You can choose from the high quality or the high coverage data set here. You predict you enter your genomic interval, your chromosome, your start stop. Um, sorry. The number of loci you want to see per page, or you can enter a gene model that you're interested in and so a flanking region. So you can either have the gene model, or if you want the 500 base pairs upstream and downstream of that region, you can enter it here. So for example, if we want to look at this gene model, you just hit load coordinates and it'll enter those positions for you there. So that's the genomic interval you're interested in. So you just want that subset of that data set. You can upload a file listing the accessions you're interested in, or you can use this kind of HTML form to enter them. So for each of the projects, we have this kind of accordion view where you can click and select the different accessions you want to look at. Um, if you're not sure what you want to look at, we provide different options in selecting. So if you just want a random selecting of 5% of all the data sets, or 10% of the data sets, or say even 50% of them, it will just randomly select 50% from all those regions. Or say you want a random 5%, but you want 50% of these Chinese land races, and say you want 25% of the Mexican land races, you can it allows you kind of do a quick sampling of the data sets to kind of get an overview of um, a representative overview of the accessions we host. So if we go down here and just click this, the create and view VCF file, of course that's not working. Oh, there we go. So what that does is it queries the HDF5 database, and it quickly pulls that information back for you. So what it does, as I mentioned, it kind of slices and dices the data set based on both the genomic region. So here's the chromosome and the position on the genome. We have the reference and alternative alleles. So the reference is what the allele is in B73. Alternative, obviously, is the alternative allele. The gene or gene models are the closest gene models that were annotated using the variant effect annotations using SNF-F, the effect type. So you can see here, there's two gene models near this variant. So you're going to have potentially multiple different variant effects on it. And then the impact, which is color coded based on what that impact is. I can show you another example of that here. So like a synonymous variant is going to have a low impact. So that's in green. Things like a missense variant, again, this actually changes the amino acid in the protein, would be moderate. Things like stop gain or frame shift mutations are going to be in red and classified as high here. And then we just have the metrics we use for filtering, and then the actual alleles at these. So each of these projects are color coded. Has a different color based on what project it is. As I mentioned, we chose some of the Chinese land races, some of the Mexican land races, and then the subsampling of some of these other data sets here. So green or 0 is going to be your reference allele. Orange or 1s are going to be your alternative allele. The gray or N is going to be missing, or there was no reads at that position for that maize accessions. If it's a heterozygous hit, it's going to be in yellow, and it's going to have an H here if it has a copy of each of these different alleles. So that's kind of the main purpose of this tool. Kind of lets you, like I said, slice and dice this data sets. Another feature we have here, another thing to mention, as you can see here, we do allow insertions and deletions. So we didn't have that in our previous version of SNPversity. So if there are insertions or deletions, that's supported here too. And then the other thing is it's linked to our genome browser. So if you click any of these genome browsers, it's going to open up that region within our genome browser. And we can zoom here just for convenience. It'll create this orange vertical line. So this is where the locus is of that variant of either that SNP or indel. So you can see it within context of our other data tracks at MaizeDB. We have about 1,000 different tracks or data sets mapped to B73V5, including the gene model. We have things like promoter regions, so you can actually see if it's 50, 100, 250, 500, or 1,000 base pairs upstream of that. So you can quickly get some context if it's in the regulatory region, exactly how far it is from the gene model. We have epigenetic data like ATAC-seq to see if this is open chromatin. In this example, it lies right on that peak in the open chromatin. We have nearly 200 RNA-seq data sets. We have things like cage data to see to support the transcriptional start sites. We have, I think, around 25 or 30 different proteomics data sets mapped um, from other species. We have TE elements. I put this on here. This is a new one we got earlier in January. So we got from the maize peptide atlas, we now have maize peptides mapped back to B73, V5. So we have a lot of different data sets where you can kind of see this variation data within the context of V5 with up to 1,000 other different data sets. Okay. Let me stop sharing again. Go back to. Okay. So that's one tool. I'm going to show one other tool where we integrate this data set. But first, I just kind of want to mention we're kind of starting to use more and more AI to develop tools and support tools at MageDB. We now have an AI data center at MageDB that kind of organizes data sets that use AI kind of separate from the others. We're doing more functional annotations using machine learning. We have protein structural tools. Lets you explore protein structures predicted using AI approaches. And what I'm going to show today is a variant effect tool that kind of predicts or scores how likelihood a variant effect is going to impact the function of the protein and how we integrate this WGS data set into it. So I'm just going to talk about this one specific thing. If you want to hear more about some of the AI tools, we are doing a workshop at the Maize meeting. So it's 2 weeks from tomorrow. We'll be giving a pre-meeting workshop at the Maize Genetics meeting. So yeah, so I'm going to talk about a tool we developed called Pan Effect, which basically predicts the functional consequences of missense mutations and lets you explore those with an online tool. So this is based on using protein language models. So if you're not familiar with protein language models, uh, they're very similar in practice to large language models like ChatGPT, but instead of being trained on natural languages, they're trained on protein language or protein sequences. Um, so essentially, I'm giving an example from the ESM model. This is from the Meta group. There's 4 or 5. There's a lot of them, but there's probably 4 or 5 major protein language models that people use for various different tasks. For this ESM model, they looked at 250 million protein sequences sampled from all organisms in UniProt as their training set. Their classification task was basically they would mask random amino acids. So this is similar with large language models where you mask random words and you try to recover those words based on the context of the rest of the rest of the sentence or the rest of the words around it. This does the same thing. So you mask random amino acids and you try to recover or predict what those amino acids are relative to the rest of that sequence, right? So they build neural network, try to minimize that loss function, trying to recover what those amino acids are. They end up with a 650 million parameter neural network, which is rather large. It's not as large as some of the LLMs that are out there, but it's a pretty general purpose model where you can do a lot of different or interesting things with. And one of them is predicting these consequences of varying effects. So what you can do, or what we did, you can take a protein sequence. In our case, we look at all the protein sequences within the B73 proteome. You run it through this ESM model, and what you can get is these log ratio scores at each positions for all possible substitutions. So in this example, at position 273, there's amino acid R, and say we want to know the likelihood of a substitution of amino acid H. So what you do is you take the probability of the residue of a H at position 273, again, within the context of the rest of that sequence. And then look at the probability of amino acid R, which is the actual value, again, within the context of that protein. And then you can get this ratio or log ratio score at that position. And you can use these values to infer or predict if it's might have a functional consequence. And so we did this for 550 million variant effects in B73. We also ran 50 of the maize genomes we have through this. So we have several tens of billions of these variant effect scores that you can download at MaizeGB if you're interested. And so what previous studies have shown is that these log ratio scores of greater than -8 has a little or benign effect on the function of the protein, where scores of -8 or less has various degrees of functional consequence, or it could affect the function of the protein. So what these two charts show is all possible missense mutations if you're looking at the canonical or representative isoforms in B73 uh, versus just a naturally occurring one. So I guess the difference is like for each protein, there's 20 possible different amino acid substitutions at each position, but not all of those are naturally occurring, right? Within the diversity of maize, where these are the naturally occurring ones that you actually see when you look comparing proteins across different maize genomes. Um, so yeah, so what PanEffect does is it lets you explore these data sets. So as I mentioned, there's 550 million possible mutations. If you look at just the canonical or representative isoforms, there's about 273 million. If we look at the pan genome, so this is Ethy Cannon and Maggie Woodhouse from our group developed a pan genome looking at 60 maize genomes. So looking at synthetic orthologs or trying to identify which proteins or genes were similar across each of these 50 different maize genomes. And you end up with these pan sets or similar or the same genes across each of these genomes. And then we can look at what are the actual natural occurring variations. And there's about 2.3 million of those. And then we can also look at the 1.3 million of that annotation table I showed previously, where there was 1.3 million missense mutations in our high coverage data set. And we actually use this tool to explore those. So there's the final thing I'm going to show. I'm going to switch sharing one more time. Okay. So here's our variant effect tool. We have a version at MaizeDB right now. We're planning the release once the WGS datasets release. We'll release a version that actually has this WGS datasets. So what this does, you can enter a gene model or say a gene. For this example, I'm going to look at knotted one, which is like a classical gene, maize gene. So it gives you some information about the gene, it gives you information about the protein. If you want to see the 3D structure or see that uniprot, you actually want to download and see all the variant effect scores, you can do that here. We also list any GWAS-based trait. So if any trait's been associated to a SNP within that gene model or near that gene model, we list that here with the source. But here's kind of the heart of the tool here. So again, we're looking at knotted one. Along the top here, the horizontal is the position within that protein. So this is a 359 amino acid long protein. And anywhere along that line is the position on that protein. We have two tracks here. One is the PFAM domains or the functional domains. So knotted one has these 4 PFAM domains in it. We also use the ESM model to predict the secondary structure. So if this region's more likely to be alpha helix or a beta sheet is shown here, partly just to help you or give you some clues on, if there's a amino acid substitution within the functional domain, or if it's going to affect the structure of the protein, then it may have more likely to have a functional impact. But also we have these scores here. So here's 2 heat maps. One's a zoomed out for the entire protein. So in this case, 359 amino acids long. We can scroll along here to see the zoomed in. So say we center around this first functional domain. That's a 50 amino acid window zoomed in for that region. again, along the horizontal is the position on that protein. And then along the vertical is the 20 different amino acid substitutions. So obviously, there's a substitution with itself. So in this case, the actual amino acid is a A. Substitution with the A is going to be in black, is the exact match. Things scored with blue or light blues are these that have the log ratio scores of less than -8. So those are scored from dark blue to light blue. Things from green, yellows, oranges to dark reds are things that are going to have a stronger effect or stronger impact on affecting the function of this protein. And you can see those by hovering over these different cells. And so you can kind of see patterns. So even though the PFAM domains were predicted or annotated separate from this method, you can see where some of these functional effect predictions overlap pretty well with these functional domains, where any type of mutation with them could have a high impact on the function of this protein. And so as I mentioned before, so this is what you'd kind of see at MaizeGB now, but we also integrated this WGS data set. So if you just toggle with this radio button, instead of seeing all possible substitutions, so 359 times the 20 different ones, this shows the actual variation within that WGS data set. So those 1.3 million missense mutations that were annotated. So we can actually explore the naturally occurring variation and see if there's predicted impacts within this region. So it kind of gives you a way to kind of filter this data set with some, the real variation within this population. We also have a tab here where you can explore the natural occurring variation, but instead of the WGS data set, this is the pangenome developed by Maggie and Ethy from our group. This isn't a great example here, so I'm going to open up a different tab if I can get to my tabs. So this is just a different gene model. As you can see, it has one functional domain, but this shows a little more where you can see some more natural variation within the pangenome. So within this region, you can see, so basically what we did here, so we took a multiple sequence alignment within the pangene sets within the pangenome. So we have these 60 maize genomes along the vertical here. So instead of the all possible substitution, these are just the substitutions that are actually occurring within these different maize genomes. And then if they're identical to B73, they're in the dark blue. If they're different than B73, then it's scored again based on using this ESM model for how likelihood it's going to have an impact. And then we sorted them and color coded by the heterotic group. If they're stiff stock, non-stiff stock, a mixture, if they're European lines, if they're using Chinese breeding programs, we have the tropical lines, and you can, use that to see if there's maybe some patterns within some of these heterotic groups of some of this natural occurring variation and how likely that'll have an impact on it. And you can see some of these gray lines here, for example, like this vertical line would be an insertion relative to one of these pangenomes, where maybe some of these gray lines would be a deletion relative to B73 in these lines. So they're not scored, obviously, because this method really doesn't score the impact of insertions and deletions, but they can be shown, and you can kind of look at the overall, some of these structural variations within these protein alignments, along with the variant effects. So that's the pan effect tool. As I mentioned, it's available at MaizeDB. Our paper was actually just an early release of that paper is available in bioinformatics now, if you're interested in seeing that. And then I just have the acknowledgement slide that I'll share, and then I can take and answer some of those questions. So this is our core group at MaizeDB. These are the permanent federal employees we have at MaizeDB. As I mentioned, Margaret Woodhouse and Ethy Cannon developed the pangenome. John does a lot of our genome browser and our back end and front end web development. So he's currently working on getting SNPversity 2 as a web version. So the one you saw today was actually running locally on my laptop. So we're working on a Docker image. So there's something that if you want to work on it locally, we'll have a Docker image that people can download and run this locally, or we'll have a web-based version at MaizeDB. I want to thank Jeff, who kind of came up with this idea of generating this VCF type of data set and doing it on a regular basis and kind of exploring ways we can support this moving forward, like how often we want to have these releases. And Matt and Arun from Iowa State, who also were critical in this project and really identifying how to best do this, identifying which maize accessions to choose, and a lot of other different choices that went into this projects. And with that, I can look at the questions. So thank you.
Yeah, thanks, Carson. There's a couple that are already in the chat.
Okay. Um, Stop sharing, I guess. Okay. Are the data sets enriched in any particular fraction of the genome, for example, more than proportional genic SNPs? Data sets enriched.
Yeah, just looking at the number of base pairs that fall in gene models, versus intergenic, and seeing if there's any of those fractions that you were mentioning that would be overrepresented.
Yeah, so I mean, I guess I don't know at like a per. Obviously, there's more SNPs in the intergenic regions, but there's more intergenic base pairs to begin with. But I do know that it's a pretty consistent proportion across our entire data sets versus the high coverage and the high confidence ones. That's something we kind of controlled for to make sure that the proportion of these SNPs were pretty consistent at each level of the filtering process.
Cool.
Um, are the land races geo referenced? I guess I'm not sure what that means. Um,
Meaning if there's
Um,
latitude and longitude, right, Fausto?
oh, um,
I can speak to that a little bit. All the project is using is things downloaded from the SRA. I believe all or almost all of the traditional varieties being used here. So they're all published. And I believe almost all of them have lat long in the original publications.
Yeah, I know that's right for the ones that we generated, Jeff.
okay. Yeah, so I guess some of that metadata is not attached to this data set, but it is publicly available. So, and then,
That might be a nice addition to the database, Carson, if we could somehow bring in some of those data sets from the original publications.
yeah, so that's some of the input we're looking for of what other data sets we can attach to it. So it's pretty easy to do that. It takes some work to do it, each time. So like collecting exactly what metadata we want, and then including that in other builds is something we could do. And then Jeff has his hand raised if you want.
Well, actually, following on that, I was thinking of an old tool that Jeff Glaubitz had on Panzea with some of the initial, I think it was TrackMan SNPs, where given lat long of samples, it was a mapping tool that would give pie charts of the frequencies of particular alleles in particular, geographical regions. Is that something that's feasible to do for this size data set, or is that easy to produce for the website?
I would have to think about it. I mean, I think it's definitely doable. So you would basically like kind of draw like a box type of region, or you just either pick the latitude or longitude.
I can't remember how he had it set up. I think he had somehow predefined some scale, and as you zoomed in and out, the pie charts changed to represent the different scales.
Okay. Yeah, I'd have to think about it. I mean, with the HCF5, it definitely is we may have to index it in a different way, but I'd have to think about it. Because the way it's set up, it's very chunked based on the genomic regions. But if you want to sample across all the different allele, that would be a
No,
different.
this would be like I choose a specific allele, a specific
Oh,
SNP,
okay.
and it pops up a graph and shows the geographical abundance of that SNP.
Okay. Yeah. Yeah. And that definitely is reasonable. Okay. Then will the GVCF be available? Um, yeah, so that's something we're working on now. So some of these are rather large data sets. So we're looking at the best place to host this data sets and what's feasible. So that's something we definitely could do. We're just kind of making those choices right now of where the best hosts some of these data sets. So and
I guess critical for folks that want to do sort of population genetics on these data that the GVCF is available. So we have a VCF with invariant sites for all 1,500, right?
yes, so yeah, so we definitely can make that available. So we'll um, kind of release all these data sets at the same time is kind of the goal.
I'm curious, Carson, so how would folks from the community um, interact with Maze GDB if they are generating a data set and they want it to be included in the next build of the whole genome sequencing? Do they just reach out to you guys directly, or if they want to nominate a data set that they think should be included, what would be the process?
Yeah, they can definitely reach out to us directly. But yeah, so I guess when we release this, I suppose that we could put a link to if they rather just enter it through a form or something like that, or through a feedback if they want to give us a list of the accessions or gives a heads up if they're generating data sets, that we can include that in future builds. Um, so that's
Yeah,
kind of we're working through and like how often to do it. And I'm not sure if I'll have it. Hopefully we'll be able to estimate like, hey, spring of 2025, we'll have a new build. So if you get us data sets by an X date, then we can consider it. But those details aren't quite figured out. But yeah, we can come up with some mechanism. Definitely, like you can email us anytime with any um, questions or suggestions, but we can maybe put a more specific link where people can enter this information.
yeah, I think that's a great idea. Like on the page for the data set, and something like don't see the data you want here with a question mark, submit recommendations for inclusion here, and someplace where they could, type some text in or something like that. And we talked about this and tried to come up with a pretty representative data set, across different interests. But a lot of the folks here, and we have very evolutionary interests. So you see land races and teosynthese and stuff in this data set. And there might be stuff that's missing that the community would be interested in.
Yeah. Pipeline. Okay. Will the pipeline be available to join with unpublished data? Um, yeah, I mean, we'll try to make the pipeline as um open as possible. You know what I mean? Like the details on how we generated this data sets.
Think
And we don't,
Marcus, maybe what Marcus is asking, and Marcus, maybe you can clarify. Are you asking if there'll be a tool that's provided to bring in a different data set and put it on this coordinate system or join it with the data set?
Yes, so if someone sequences a whole new data set and wants to join into this data set and not just wants to join the SNPs, but actually do the whole calling, and then afterwards that would also make it easy to, contribute to the data set, but not everybody might want to do that before publishing it. To do the same calling basically, but with additional accessions or material. Is that feasible, or is it any way
uh,
computationally impossible for anybody else than you?
I'm not sure. Yeah, I don't know if there's an easy way to take data and do new callings without doing a whole new build. You know what I mean?
But
Um,
basically what I'm saying is basically doing the whole new build. But is that feasible, or is that anyway not something that makes sense?
yeah, I don't know if that's something we would offer. Um, yeah, that's what, as Jeff mentioned, we'll definitely give you all the details on if someone wants to do the build themselves. I don't know if we would be able to host that beyond what we plan to do on our regular builds. You know what I mean? If you want to incorporate into the next build, that's possible. But if you want to run on your own, then I think you would have to, and we would definitely help out if you had questions on any of the choices we made.
Yeah, and I think if folks are bioinformatically savvy, they could replicate what GenCove has done. And it might be worthwhile, Carson, just to post your code, on GitHub or something like that, if you haven't already, for doing the filters on the data set, just so that people could filter their data sets in exactly the same way.
Yeah.
Awesome. Thank you.
Other questions for Carson? Okay, well, thank you so much, Carson, for a great talk and for putting this resource together for the community. You guys will see there's an opening for next week. So if you'd like to give a practice talk for the Maze meeting or anything else that you'd like to talk on, please let me know in the next day or two. And I'll let folks know early next week whether a talks materialize. But otherwise, we'll see everybody hopefully at the Maze meeting in Raleigh in a couple of weeks. So thanks so much. Enjoy the rest of your day.
Thank you.